Okay. All right. Hi, welcome everybody. Thank you so much for coming to our lecture today on this beautiful Wednesday. And I welcome you from my home and also from Dr. Schwartz's home as well. So the two of us, what we'll be doing today is that we'll be presenting to you viruses. And we're really going to go over the ultimate basics and nuts and bolts of viruses, more or less from a different perspective. And because we are here in this global pandemic, we're all at home because of a virus. And the thing is that I know that we all have that background in microbiology from either high school or from undergrad, and and all that kind of stuff. But we all have that from a different mindset, and from a different mindset as a student. At least myself, when I was a student, all I did was memorize as much as I could so I could get the higher grades and all that. But I didn't really get the chance to really understand it from a real life aspect. I would say that now that we're in that global pandemic, honestly, as I was preparing for this particular lecture, I found this to be almost a refreshing refresher on viruses. So let's talk about the nuts and bolts. And of course, what we will be going over will be the ultimate aspect of COVID-19. And we'll go over some recent data that is out there. So nuts and bolts. Here's my little one. Here's Anderson. He's 4 years old. I mean, we're not going to go as nuts and bolts as this, but I know that most of us are homeschooling our kids right now. But here's Anderson, and funny enough, about 2 weeks ago, he was assigned to create an ABC or an alphabet book. So I gave him full reign as to what kind of words to associate with each letter. So of course, A was for apple and B was for box. And then we got to I, and I just had to show you guys this, but I know it's not a word, but he got to iPhone, which I thought was kind of funny. He's a 21st century child here. And then he did get to Q. Q, his first word that he came up with was quarantine, but I think I, as a parent, will probably get in trouble by the teacher if he started drawing, a poor child behind bars for quarantine. So he came up with the word question, and then we got to V. And maybe it's because he's hearing a little bit of this around the house and all that, but immediately he went straight to the virus. So this is the rendition of a virus from a 4-year-old. So what is our lecture about? I mean, overall, just letting you guys, this will be a 1-hour lecture here, but we'll review exactly what is a virus from the most basic aspects, how the virus works, and of course, we can't not talk about COVID-19. So what's a virus? I mean, when we think about viruses, there are all these different kinds out there. There's HIV, there's the MERS COVID virus, there's the varicella virus, Zika, herpes simplex virus, especially we encounter that in the dental world, and of course SARS-CoV-2. So there are different kinds out there, but really, truly, the main thing that draws them all together is this. This is what I see when I think of viruses. I think of the ultimate killing machine parasite. Maybe because I'm around little boys a lot at home here, but this is what I found, and this is what really resonated with me. So picture a lot of these things in your system. Picture a lot of these things taking over the entire planet. Parasites, that's really what technically a virus really is. And of course, I'll go more in detail about that. But really, how many is there? So let's do a little bit of math here. So let's take 1 billion. And let's multiply that by another billion, and let's multiply that by 10 trillion. And here we go, we have 10 to the power of 31. So what does that mean? There's this many zeros before the decimal place. And if you take every single virus, all of these things, and you line them all up, how far does it go? It goes approximately 200 light years out there into outer space. So that's a lot of little viruses that are out there. But random fact, most of these viruses are found in oceans, thank goodness, but they could be quite deadly. But one may argue that viruses is the most abundant life form on Earth. Is it a matter, is the virus really a life form or not? It's really quite debatable there, and I will explain why. Afterwards. But the next interesting fact is that 5 to 8% of the human genome is viral. That blew me away when I was researching for this particular presentation. It's just that there have been ancient retroviruses that have coded into our genetic code over the generations, which to me is rather creepy and interesting at the same time. So as I mentioned, like, is a virus life or not? Well, viruses themselves are the bare bones. It is really the borderline of life. They don't grow at all. They don't reproduce on their own. They have absolutely no metabolism. They're just kind of there. They're really just the shell. It's the shell with a lot of, with nucleic acid in there. So there's just information about the virus. They're fully dependent on a host. So is it life or not? Well, it does depend on living things. It's a huge debate altogether. That's for another time. But viruses are truly the bare bones of it all. What's really important is that, well, if they are really the bare bones and they're out there creating all this disease in the world, they have to pack really lightly. So they're very efficient at getting from one cell to the other, at one being to the other, one animal to a human, human to another human. They have to pack really lightly. So they only have to take with them what's really necessary, and this essentially is the genetic code, and just something for it to hook on and grab onto a host cell. So these are basically killing machines that do take over cells. So they were the ultimate brainwashers. They would take over a cell and change the way the cell is and revert them over into virus-making machines or factories. And for other viruses like HPV, that could turn cells into cancerous cells, like in the sake of oropharyngeal cancer. So here's the really, really basic diagram of what a virus is. And honestly, as I was going through this whole thing, I forgot really what was the main foundations of what a virus is. There are really three main components, 2 to 3. The reason why is that not all viruses have an envelope, but in the case of the COVID-19 aspect, the envelope is actually a lipid membrane that is surrounding it. And so there's the envelope, there's the capsid, and there's also the nucleic acid genome. So what's really important and interesting to me is that the envelope is a lipid membrane. As creepy as it sounds, this lipid membrane is not derived from the genetic code of the virus itself. It's taken from the last host cell that it took over and hijacked. As it's almost maddening to almost hear about that, because it just takes everything and brings along its cells and inserts into another cell and then replicates again. But what's interesting in terms of public health is that, thank goodness, this COVID-19 has a lipid membrane. Why? Well, things like soaps and detergents can, because they're lipophilic, can degrade that lipid membrane and inactivate the virus. There are other things that do degrade a membrane, and of course that's hydrogen peroxide or bleach, for example, in certain aspects of UV light apparently. So there's the envelope. So the next one is the capsid. So picture almost like a bike helmet on protecting the nucleic acid or the genetic code of the virus. It's important to protect it against nucleases, which are enzymes that degrade that nucleic acid. And it also, this capsid contains receptors that help bind onto the particular virus. And they come in different shapes and sizes, but at least for the sake of this particular lecture, we'll just make it nice and basic, just like this. So finally, really the gold of it all, which is the virus's genetic code, it's nucleic acid genome. And this either comes in one or two things. It's either there's DNA or you'll find RNA in the virus. And this particular genetic code codes for the virus itself and sometimes the polymerase, which is the enzyme that's responsible for the gene, for the transcription of this code there. So before I go on with viruses, the one thing that really comes into mind is, well, how does that really differ between viruses and bacteria? So let's go back into memory lane. The major thing is that bacteria have everything to live on their own. They are single-celled living organisms. Let's go back to the virus. They're not single-cell living organisms. All they have are just these three things attached to them. So it doesn't have all the stuff that makes it to live. So let's go back to the bacteria. It has all the stuff. They're not parasites. They can live on their own. They have the cytoplasm. They have the cell membrane, the cell wall, the capsule, the mitochondria, the nucleoid, and the plasmid, and something that makes it propel and move from place to place, like pili or flagella. Because it has all this stuff that is embedded in the bacteria, they are much, much bigger. The studies show out there that bacteria are approximately 1,000 nanometers, as opposed to viruses that range between 20 to about 300 nanometers up there. Okay, so that's those are viruses right there, and of course bacteria. So how does it work? Well, viruses need to somehow travel from one place to the other. There are different modes of transmission. First is contact transmission, either directly from a person with a virus and transmits it directly to someone without the virus who is healthy. So that's direct from person to person, or it could be vertical from mother to baby. And the other one through contact transmission is the indirect transmission via foaming. So you would have a patient with the virus somehow coughs on a table or a surface, and then a healthy person walks by, touches that surface, and then touches their face. That surface that's in itself that is contaminated is called a fomite. So that's indirect contact. And what's interesting to see is that there have been studies that shown that we tend to touch our faces every 3 minutes. So at least in terms of COVID, it's something we should really stay away from as much as we can. Other modes of transportation or transmission of viruses includes vehicle transmission, that is right here. So vehicle transmission is done it's either transported through water or through food, or in the dental world, it's aerosols. And then finally, vector transmission through bugs. This is not in the case of COVID, of course, but something like Zika virus that's transmitted through mosquitoes. So there's no way I'm going to go over in detail this, but this is in general the virus life cycle. What happens? So when you have a virus here, it has to attach or dock onto a receptor of the host cell. So this whole thing is the host cell. And then the host cell has to somehow kind of get into itself. So this is called endocytosis. And so that the cell membrane just wraps around that and just basically spits right into the cell itself. And then that's when the machine begins, and that's when it starts transcribing all these things. It transcribes the genetic code, and transcribes in a huge factory the proteins for the capsids, and then they put these things together back into the exact same copy as the virus that came into the host cell in the first place. And from there, it has to get out of the host cell and then go on and find someone else to infect. So This is what I think of, is that viruses are evil. Here's the coyote from Bugs Bunny. Viruses are evil, I think, because once they infect a host, it takes over that host cell completely. It changes them and hijacks them and changes them into virus-making machines. The original function of that cell is completely eliminated, so they don't do what they used to do before, and they're forced to really go ahead and just produce virus. And after that, in order for the viruses to get out, it has to kill that cell. So they're murderers in addition to that. All right, so those are the nuts and bolts of viruses. So let's talk about COVID-19. Well, okay, before we talk into COVID-19, coronaviruses. So a coronavirus is a type of virus that is very particular. What is really unique about that is that if we look here in this picture, we see these projections, and these projections are quite characteristic for the coronavirus. It creates almost like a crown around it, which I'm sure you've all heard on the news and on media and all that. So that's why it's called the coronavirus. It's almost like this dryer ball here. My 4-year-old Anderson, the first thing when he looked at this, he goes, "Oh, mommy, it's a virus." And he throws at me, he goes, "Ha, you have the disease." So here is a virus. So it has all these projections, and these projections are very important for the spread of disease. But in general, coronaviruses, it causes diseases in mammals and also in birds. Okay. The disease or the virus du jour, COVID-19. Well, COVID-19 is the disease name. So it's an acronym for Coronavirus Disease 2019, because it was in 2019 in December where they found that this disease was in humans in China, in Wuhan, China. And then the virus name for the exact same thing is called SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. So if you see those two names, they're exactly the same thing. They are interchangeable. So let's do a little bit of epidemiology really quickly. So I checked right before we logged on just now. So as of 11:30, there have been 3.1 million, there's a zero at the end of that, sorry guys, zero. There's 3.1 million confirmed cases of COVID-19. And as for deaths, it's 225,000 cases as of just about half an hour or an hour ago. And if we bring this home a little closer in Canada, we have now about 50,000 cases that are confirmed, and we're almost reaching about 3,000 deaths right now in Canada. The reason why I highlighted Ontario and Quebec on the side here is that Ontario and Quebec are the ones who have the most cases. And note, these are the confirmed cases out there. Note that there's a lot of variation on testing between the provinces. They have very different testing strategies. In Quebec, they're testing everybody in nursing homes as opposed to other provinces. So this should be taken with a little bit of a grain of salt. So it doesn't mean everyone has it. It's the ones that have been tested, and not everyone tests the same way in each jurisdiction. But the one thing you can't argue with is death. So that's pretty black and white. So deaths in Ontario, there's 34% of all deaths in Canada is in Ontario. And then as high as 58% of those who have died in Canada is in Quebec. So here in Ottawa, as of 1:30 today in Ottawa, 1,297 have confirmed disease, and then 74. Sorry, I have something in the way there, but I think 74 deaths have occurred as of 1:30 today. And so let's go down more to the basics again, back to the microbiology aspects of stuff. So what is really the key for this COVID-19 to be passed on to various host cells and transmitted out into the public? The biggest key is the ACE2 receptor. When I first saw this, when I was researching for this, ACE2, it is, what do you think it is? It's the angiotensin converting enzyme 2 receptor. Or if you remember back in the renin and angiotensin system there that regulates blood pressure, well, that's it. And it's really this, it's the S protein or the spike glycoprotein that is found on the envelope or on the outside aspect of the virus that docks on to the ACE2 receptor. So where is this receptor found? It's found in the lungs, it's found in the kidney, and it's also found in the GI tract as well. So what happens if you catch it? Like how do you get it? So there's direct transmission through respiratory droplets, indirect transmission via fomites. Someone sneezes on a surface and then someone picks it up and touches it and then touches their face, and also aerosols. But basically once the virus enters into the nose and throat area, it latches on to the ACE2 receptor on the cell surface, and that's when everything starts. That's when the viral replication happens. And it takes time for that to happen before anyone gets symptoms. So there's an incubation period. It has to cook for a little while before anyone gets any symptoms. And you have to develop some sort of viral load. You have to develop a certain amount before the body really, before anyone notices it. So that incubation period is about a week. And what is shown now is that a person can shed these viruses at very high concentrations about 2 days before the onset of symptoms. So this cannot underestimate the importance of hand washing, and of course the social distancing and all that. But 2 days beforehand, that's when the virus starts to shed and makes one quite contagious. So in the symptomatic period, so after about that week's time, one will feel feverish, there'll be shortness of breath, there'll be a cough, so these lower respiratory symptoms start. Then the patient will start to feel fatigue, sore throat, muscle ache, anosmia, that means a loss of taste and smell, and diarrhea, and also vomiting is also a common symptom on top of that. And what's interesting to note is that it's really almost day and night in terms of the presentation, the clinical presentation of COVID-19. So about 90 to 95% of those have a mild disease, and it's quite self-limiting, thank goodness. So that's why when we're looking back in the epidemiology numbers as to what is being reported and confirmed as disease, there are a lot of patients who have it and they're just at home, or people who have it and they're at home and haven't been tested at all. So the number is there's a bit of a range out there, is more of an estimation, but it's about 90-95%. But it's those who are quite unfortunate, and it's the 5 to 10% that require hospitalization. And who really is at risk of getting to that extent that they have to go to a hospital to be treated. And it's really the elderly. So it's those who are about 70 years and over, and also those who have comorbidities like chronic lung diseases, obesity, hypertension, diabetes, for example, cardiovascular diseases. Those are comorbidities, and who are older are generally at higher risk of being hospitalized with COVID-19. So what are the main targets? The biggest one is really the lungs, as you all know. So once it reaches down into the lungs, it goes straight to the type 2 pneumocytes, and these are the cells that line the alveoli in our lungs. And the alveoli are the air sacs. They're the things where it's responsible for exchanging the gases of oxygen and carbon dioxide as we breathe. It's certainly a very important thing. And once that's compromised, then we're at very high risk of hypoxia. And if that infection is still not controlled, then the alveoli start to malfunction. They start to fill with mucus and debris, they get infected with bilateral pneumonia. Obviously, you can imagine the transfer of oxygen and the exchange of carbon dioxide from the blood is quite poor. And patients are just having problems with breathing, and their lungs are just overworked. And the unfortunate few that keep going downhill downhill experience acute respiratory distress syndrome, or what's called ARDS, and about 2 to 3% of those they may experience that, and that's when they need help with breathing. So they need intubation and ventilation, and often these cases don't do too well, and most of them are quite fatal. Other than the lungs, other places that affects would be the coagulation system. So there's an increased risk of thrombosis. So even in younger patients, there's an increased risk of coagulation. The blood just gets thicker and thicker. So they're at higher risk of embolism, of stroke, and of a heart attack as well. The one thing that's quite interesting is, for the really unfortunate few, is the cytokine storm. It's just that when they're attached by this virus, all of a sudden the immune system goes on haywire. It just goes nuts, to the point that the immune cells, or the white blood cells, start to spill out so much of the cytokines out there, which increases the inflammatory response and thus ends up killing healthy cells. There's some evidence that the body tries to somehow accommodate for that, to release those cytokines into the system, where the blood vessels start to thin out a bit, to allow those cytokines to come out. And so as such, there are these little holes into the blood vessels. So what does the body do to repair that? Well, it has to coagulate and patch up those holes as well. So that leads in another mechanism for hypercoagulative state as well. So when you're losing a little bit of blood to the organs, what happens is that you end up with multi-organ failure, and ultimately would be death. So that's the cytokine storm in the minority of cases. So we have the storm, but we have people that are asymptomatic, so no storm, so sunny skies. So about 25 to 50% of those are unaware that they even have this disease in the first place. And there's a wide variation, or wide range, on this prevalence. The reason why is that the studies are mostly done on the cruise line ships, so the Princess Cruise Line, where they've tested everybody. So they knew about between 25 to 50% of those cases were tested positive for the disease, but they had no idea they even had it. And then also some of these studies were done in nursing homes as well. So it's not done in the general public. There's a bit of bias, and that's another room for discussion about that, but obviously there's a room for bias for something like this, and of course the N is small. But there are people walking around that have the disease and have no idea they have it. So physical distancing, social distancing, doing our conversations and our courses like this is incredibly important. And from there, I wish you all well, and I'm going to pass on the torch, this little virus torch, over to Dr. Schwartz here.
I don't know if I did that the right direction or not.
Hey guys.
Hi, everybody. Hi, Karen. Okay, so we're going to try and share my screen. So, Karen, if you could take yours down for a sec. Stop sharing yours. And here we go. Okay. So a couple of things before I start. Here we are. I'd like to first say thank you to everyone for being here, or being there as I'm here, and tell you I'm actually a little anxious about this one. For one, it's not easy to follow a brainiac and such a clear speaker like Dr. Fung, so I hope I do you guys proud. And secondly, although I'm used to talking to myself, I'm not used to doing that in front of other people. And the other aspect is, I'm not a virologist, so I've been intimidated by my ignorance in this. Over time, though, I've realized as I've been reading that I'm not the only one, and even experts are feeling ignorant. And one of the reasons is things are changing so quickly. I'll tell you right now that whatever numbers I have in here are wrong. The stuff I'm going to talk about, that's more confusing. And you see that things are changing daily. Last week, I gave Karen all the slides, said, done, I'm good. And every day I've changed a bunch of slides. Last night I just stopped reading because things are changing. as I was getting ready, I looked at my phone and new information had come up that could have potentially been relevant. So the goal here is really not to be exhaustive, but to really distill whatever information is there, kind of for the basics. So you can apply these principles as you're reading the ever-changing articles, because it's literally every couple of hours things change. My best example of that is we're going to talk about the viability of the virus, and there was an article, a big one by the New England Journal of Medicine, and the month later, that was in March, April, they already sent an addendum, and things change. So everything is changing really quickly, and it gets confusing. So hopefully these building blocks will help us figure out how to take the information, the wonderful base that Dr. Fung left us with, and how are they using that information to move forward. So we're going to focus on the technologies and things that are going to help us get on the road back. So as Karen has showed us, we've gone to the point where we've gone through the clinical picture. I'm going to start talking about testing, vaccines. I'm going to talk a bit about virus stability and go over some terms, but I'll do some as well as myself. What's unfortunately lacking from this is treatment. We don't have treatment here. And one of the reasons I'm not talking about it is A, we don't have a lot of time, but B, there aren't any major gold stars for runners. I do have a slide on chloroquine, it's in the news all the time. And literally, as I was picking up my phone, I think the FDA just approved one for trial, but there's no real magic bullet thing that's running ahead like there are in the other aspects. So let's start with the testing. So a couple of things before we start talking about the testing is the countries that have done the best have been the best at testing. And there's a few reasons why they're in that situation. As you know, most have been through it before, Singapore and Korea and such. And so they have a bit of infrastructure. They also have the fear in them because they saw what happened the last time. But one of the key things they have is a government that realizes you have to change the legislation. We have all these checks and balances in place to make sure, things don't get out into the medical field that aren't tested enough. And they've realized you've had to relax some to allow more to get in. And so I don't know if you followed it in the States, the FDA initially didn't relax anything since things were strict. And so there wasn't enough tests, and they couldn't get them to market fast enough. Then they lowered it, more came in, but then you had ones that weren't efficient, and now everyone is finding a balance. I'll talk about the interplay between industry, science, and government a few times during this, but I can't overstress the importance. So back to testing, there's basically, in rough terms, two types of tests. There's some that are based on the genetic components, the nucleosides, the RNA, and those proteins, and there's antibodies. And I'll talk a bit about both. The PCR is the main way historically of testing for the virus, because it's testing the nuclear material, the genetic sequences of the virus. It's more certain, but it takes longer. You have to have an expert person take the swab. The swab has to go to a lab. The lab does it, and then you get back. A lot's being done with PCR to make it faster, but it's just not there yet. At the time of the initial writing, again, everything changes. The FDA and the World Health Organization only really suggested those should be looked at. You hear a lot of talk about antibody testing. It's a lot faster. I'll go into a bit of detail in the next slide. But it's not really great for deciding who has it or not. It happens later in the infection. What it's great for is looking at, has the population been exposed? I'll touch on this again later when we talk about vaccines, but one of the feelings is that if people have had it, then they should be immune. And we're not 100% sure on that. There's anecdotal evidence that some people have had the condition and then have been reinfected with it. So you have to use this, again, it's a changing field, and this is moving quickly. So in terms of antibody testing, they're looking at a couple of different types of immunoglobulins. That's what our body mounts as a defense against infection. There's IgM and there's IgG. IgM happens earlier. Some people looked at it. They're not looking at it so much. You were able to get a much faster test with the antibodies than with the genetic sequences, but they're not worth as much. The IgM, it takes a few days for it to build up in our system. So you could be the most infective, as Dr. Fung pointed out, and still not show up as having it if you're doing an IgM test. The IgG tests are what they're looking at more because that should show what percentage of the population has it. Germany is leading the world with this, and they're testing virtually everybody for, or as many as they can, for the IgG to see what percentage of their population has been exposed, so they have the information with which to deal. One of the concerns in terms of whether you have immunity or not after might relate to how much is there. So again, there's so much variety in the types of tests that people use that could be relevant. And ELISA, the enzyme-linked immunosorbent assay, is one that might have a better sense of how much is present in the body, and that might be relevant. So the challenge to get the tests out, there have been a few of them. So one of them I talked about is government has had, rightfully so, legislation and rules to make sure stuff doesn't get out that's not ready yet, and they've had to relax those. The biologists are looking to develop what are we testing, and we're lucky now because we're at a time where we're ready for this more than we were beforehand. I'll stop for a second and say I found a lot of hope in reading this. When you look at the depth and breadth of brain power being thrown at this, it's staggering. And we've had a few things in play that have allowed this. So for one, previous epidemics in other places have helped, but we also have these supercomputers that are relating to reading genes and determining gene sequences. And that's been huge in determining what we need on this virus to target, whether it's for a vaccine or it's for a test. So in one sense, the biologists are rushing to determine the best thing to test. At the same time, you have to get a bunch of tests out. And firstly, there's supply chain issues for testing because there's reagents and things that they don't necessarily have enough of. And the other aspect is you need to be able to scale up to sell or produce millions of them. And we don't know which test yet. So you have these engineers working with factories to try and develop tests before they even know what test is going on. So the amount of communication going around is incredible. And different companies are starting things before they even know if it's gonna work or not. So to go over a couple of types of the tests that we're gonna start to see, one of the main things we need in terms of getting back to dentistry as soon as possible is a point of care test, because the other stuff is too slow. You would ideally like society to have stuff that comes quickly, and even before we're all ready, if we can get it in our office, that would be wonderful. So there are a lot of different tests. A lot of the stuff is a proprietary, so you don't get a lot of detail necessarily on what it was. But there are kind of three main things coming up, and I'm highlighting the Canadian companies for obvious reasons. But one that has promise, it's not accepted yet, is a lateral flow assay. So lateral flow, I'm not going to get into the physics of it just because we don't have the time. I don't really understand it. But that's one way of looking at it, and you can look at all different fluids for presence of the virus. LAMP testing is something that you're reading a lot about. It's been approved, and it's also a point of care type of test, so you can get it quickly. The one that impressed me the most was this one, not just because it looks like it was designed by Apple, but it's a point of care test. It's being produced in Canada. It has already Canadian approval. It does one test at a time, but you can see how in an office you can easily have something like this. The issue is they're being sent now to outlying communities because that's where it's hard to get the samples and the tests and stuff like that. So I think testing is on its way. We're probably, maybe a couple of months away from seeing it as widespread as we like, but there's hundreds of companies working towards it across the globe. I'm proud that we have some good ones in Canada that are moving forward with acceptance, and this would be one. Okay, so moving on to vaccines. I'm going to go through it. There's a couple of things to go through it. Firstly, if after all of this, there's going to be a Big Short type of movie where you see that there is one person who was onto this before. The name is Sarah Gilbert. I don't know if it's going to pan out this way, but she's at Oxford. Oxford is kind of leading the way in terms of vaccines, and they're working with the chimpanzee model. And she's going back and forth with antibodies from vaccines from chimps to human. It worked well in with MERS. And there's a very cute interview where she said she woke up one morning to everybody offering her money because this was the area that she was looking at out of Oxford. The other thing to know about vaccines is there's a major testing center in India. So once again, getting things scaled up, that's where things go, and the communication between all areas is fascinating. The other aspect that you want to know about is there's the Coalition for Epidemic Preparedness Innovations, and that's CEPI out of Oslo, and they're the ones coordinating this, and it needs a lot of coordination, as I'm about to show you. Okay, so what's a vaccine? Basically, it's a substance that stimulates antibody production. It gives you immunity to a specific infectious disease. It can target different proteins of the virus itself, or it can target processes. So they'll look at what the spike protein, because that's involved in attachment, that could be where they're looking at it. And again, you have supercomputers deciding which are the ideal sequences of genes to look at for this, just like you were for the tests. And basically what happens is you give someone whatever the substance is that's supposed to prime the immune system. It does that, and we start developing antibodies for it. And then as soon as whatever condition presents, you come in contact with the disease, you're armed and ready to take care of it. And that's what it's there for. So I'm going to divide this loosely into 3 types of vaccines around. There's ones where you actually have the substance, the virus in play. There's some where you have toxins from the virus, and you could target those. That's not relevant for this virus. It's relevant for other things. And you can also have subunits where it's individual aspects to it, individual gene sequences or amino acid sequences. And that's where this is heading. The PCR works along these lines, and all those individual tests works along those lines. And the vaccines are going to be working along that too, because as you'll see later, it's easier to target that. So the live attenuated vaccine is the one that's been the standard. That's how it started. You get a weakened form of the virus or bacteria, and the immune response is just like you got it, and you fight it. And then usually you're immune with 1 dose. The downside on this, or the risk, is safety, because you're literally injecting the disease into someone. And so if they're immunocompromised, if there are other issues, it could get serious to the point that they could even die. The inactivated vaccine is a safer version. It's just less effective. And when you had boosters before, it's because you've had an inactivated vaccine and you need multiple boosters to get them done. So those are the nuts and bolts of those vaccines. I'll talk about the protein aspects to it a bit later. So how do we get it done quickly? Typical development is 10 years. Ebola was considered 5 years. And so it depends on what type of vaccine you have. There's a real problem in terms of using actual virus, and my guess is it's not going to be the first one, because primarily it has to be fermented and cultured. So 1, there's scale and time issues with it, but 2, you need, I think it's called a stage 3 type of lab to be able to house this stuff, and you can't just keep this virus anywhere. And there's very few of them around, and B, as you want to scale up, you can imagine the more controls on a place because of the danger within it, the harder it's going to be. And again, thankfully, we have these computers that are able, and the people studying it are able to say, there's specific sequences of the genome that they want to target for the vaccine, and that's what's making it designer, and that's what's making it much, much better for all of us. I'll talk in a second in terms of what to target. Again, in terms of getting it out fast, clinical safety is a big issue. One of the things that the regulators are trying to work with now is seeing how fast we can get to test. Usually testing on humans is a long way out. It's like last after in the 10-year process. They're trying to figure out how and when to get this stuff done faster than they are. I think that Oxford study is going to be testing in February, from what I'm understanding. So, and again, the production and the regulation are things that have to happen hand in hand. So normally when you develop a vaccine, Okay, I'm going to take it a step back and say, as I was looking at this, I had a fear, and you might have some of the same fears. So I figure I'll answer this question now. We don't have vaccines against all viruses. HIV never had a vaccine, and herpes never had a vaccine. Those are the ones we know about through our work. And go through this panic. What if we never get one? And I looked it up, and there's a lot of differences between the two of them. Sadly, one of the differences, to get a vaccine, you need to recover from the disease. You actually never recover from HIV. Once you get it, it kills you. Very, like, there might be a couple of cases, but no one's successfully fought it off. So you don't get that IgG out of the case. And so that's one part. The other part is it's tricky, and it inserts itself into the human genome. And so it's very hard to target an antibody against us. Anyhow, going back, which vaccine, how do you determine which vaccine to do? Normally what a company or a university would do is you look at multiple candidates, you go in sequence, you try one, does it work? No, yes, try another one. And usually there's a lot of failures before you find the ideal one. What's happening now, though, is we're working in parallel. And this is what that CEPI is organizing. All different labs are looking at different sequences as determined by the computer, and they're determining which ones to target. It's much more expensive, but it's being, the cost is being put across a whole bunch. So January 11th was when we had the sequence in hand, and that was sent out. Again, every number I have is wrong. 78 different groups were looking at April 8th. There's probably hundreds. For the information, most are looking at viral uptake proteins. As Dr. Fung talked about, that's what makes this one special, and that's how it attaches. And 70% of them are from private firms. So it's interesting, it's great. You can't always get their information, again, because it's proprietary for obvious reasons, but that's the lay of the land when it comes to that. A term you hear all the time is convalescent plasma. Convalescent plasma is what they're taking from patients who have recovered. So ideally, you've recovered, you are now immune to it, although, as I said, we don't know if that's true or not. That's the hope. But there are definitely anecdotal cases of people who've been infected and have not had immunity after. So this is something that definitely has to be looked at, and this is how. So the Canadian version of it is Concord. There's a whole bunch of countries that have their own, but there's a lot of study going on to convalescent plasma to see if it can be used to help determine immunity. So on to the treatments. There is an antiviral that's looked at, remdesivir, but it's not there yet, and you've been reading conflicting things. The one I thought I would talk about is chloroquine, just because it's in the news. There is a bona fide reason to look for it. Early evidence from France and China showed it worked. It definitely, in vitro, in petri dishes, can kill the virus. It's used in combinations with azithromycin. It was used for malaria that we knew. I didn't know it was also used for inflammatory conditions. The downside is in vivo, it can provoke dangerous arrhythmias and make things worse. So the FDA is recommending against using it, but you shouldn't really be using it off label for that yet, because they are still testing it. So it might have some promise, it's just not there yet. So virus stability is something that is obviously really important for us. Again, as of now, there are two basic studies that have looked at it. One in the New England Journal of Medicine that I alluded to earlier, and the other in Lancet. The New England Journal of Medicine came out with two from the same group because they did an addendum and got it rushed out. So in terms of temperature, the fridge is an ideal place for it, 4 degrees, and they found it active after 2 weeks. Whereas at 70 degrees in Lancet, they found most of it was inactive. So that's favorable for us. Again, these are laboratory conditions, and how it works in real life is potentially a little different. And again, more studies are going to be coming out. They're working on them now. I just put this all together. The two of them looking at the different substances. Ideally, you see that there's a bit of difference between the two. Again, it's the way they were measuring things. Stainless steel and plastic are longer. For some reason, it tends to be more infective on smoother surfaces. So initially, when they were talking about the gas pump in Ottawa as a, you know, a potential way you can get it, it's true. It was cold. It's a smooth stainless steel surface, and that tends to really favor this virus. They tested surgical masks for obvious reasons, and for some reason they were able to find that even though it's not consistent with the other findings, it tended to last a good 7 days on the surgical mask. So as Dr. Alibi can attest and talked last time, there's almost a choreographed way you get into and out of your PPE because it's on the surface, and at least on the masks, you know it's viable for a long time. I just put the graph in to show you what the rate of decay looks like for the different ones. I took this from The Economist because the one in the New England Journal of Medicine it was all one big one. It would have been hard to see, but here you can see the way it goes down. This is our big issue. Their droplets less than 5 micrometers is considered an aerosol, and they found it viable in aerosol for 3 hours. It was still viable. I forget if it was Lancet or New England Journal of Medicine, but it was viable in the aerosol 3 hours later. So that's possibly where the college is coming from. Whether it's suspended or not for the 3 hours is a different animal. I just read now about there is a dentist in London who's using, I forget, it's a pneuma, anyways, it's some form of vacuum that they use to keep hospital rooms sterilized, and they're trying to work with that. Again, that's not right up close to the mouth, but you know, maybe there are ways to get the room cleaner after and decrease that 3-hour wait that they're looking at us, and maybe that's the type of thing that we'll be looking at. The bigger issue for us, obviously, is the proximity, and we're literally nebulizing water and activating the generation of aerosols. So just towards the end, Karen already alluded to this, virus shedding. That's the release of the virus. I need to look some of these up, so I figured I would put them out here for you if you're reading stuff and not completely sure. So it's the cell releases the virus, and the virus goes to either to other parts of the body or it goes into the environment. A fomite, which is something I didn't know or had long forgotten, is objects or materials that are likely to carry the infection. So you think of the gas pump handle for that one. Herd immunity is something that's getting a lot of discussion now for obvious reasons. It says half, it's more 60 to 75% of the population to have it. They say for it to stop. Picture 5 people in a room, 4 of you are immune, 1 person has it. It's not going to leave the room other than through that 1 person. You're not going to spread it so you have it. So that's where they say the herd immunity is from. As you know, Sweden is experimenting with that. They have a higher death rate than the countries around them, possibly because they're just letting it go around, and it has to be combined with other distancing measures, and you know, we have to all figure that out as it goes. So there's the demics. Just to review, the epidemic is when you have a lot in an area. A pandemic is when it's spreading over multiple areas. Endemic is when it's found in a particular group of people, and academics, boy, do we need them now, because they're very helpful. So again, as we come to the end here, doing this, A, confused me, but B, made me feel hopeful. You know, you're literally reading article after article on just all the innovations that are coming around across the world. The fact that we're able to dialogue over the internet, the fact that information is shared at breakneck speed, the fact that all this stuff, the best and the brightest are working on it. And if you want an article that shows that, it's dry, it lists a whole bunch of them out, but you just see the scope of stuff. And if that was on April 24th, double it, because it's moving at a breakneck speed. So there we are. I'd like to give a couple of thank yous. Obviously, first, Sean for organizing everything for us all the time. Karen for being awesome to work with on this, if not a little bit intimidating. I'd like to thank everyone at Gumdocs, who I miss dearly, in particular Dr. Jones. You know, hard times call for hard situations, and you see who stands up tall, and he has done so for the office, but as well for the city in ways many people don't know. And along those lines, I'm going to indulge myself 1 more minute. And you never know how organized dentistry, what it does for you and how important it is. But I've watched people at the ODS, notably Michaela Fitzgerald, J.D. Blau, supported by Claire and Catherine, work their tails off, making sure emergencies are cared for, making sure we have the PPE, just taking care of everything. And I cannot say more how impressed I am with what they've done, and we should all be grateful to them. Thank you very much.
Yeah, thank you very much, and we really hope you guys all stay safe, and of course, if anyone has any questions, they can most certainly forward them out to Sean, and we'll be more than happy to answer them all for you. But we really hope to see all of you soon, and we'll see you on the other side.